This method, CARIC, can help answer key questions in the field of post-transcriptional gene regulation by providing a comprehensive synthesis of RNA-binding proteins.
The main advantage of CARIC is its ability to capture proteins bound to various kinds of RNAs, such as mRNAs and non-coding RNAs.
This method can be used in various cell types and organisms to study the RNA-protein interaction network.
First, culture HeLa cells in supplemented DMEM at 37 degrees Celsius in a 5% CO2 atmosphere. When the cells reach about 80% confluence, replace the culture medium on each dish with 15 milliliters of pre-warmed fresh medium. Add 15 microliters of 100 millimolar EU to each dish to a final concentration of 1 millimolar. For experimental and no UV control samples, add 7.5 microliters of 100 millimolar 4SU to each dish. Cover the dishes with foil and culture the cells for 16 hours using the previous conditions. After this, add half of the previous amount of EU and/or 4SU to the appropriate plates, and continue culturing for another 2 hours. To begin in vivo cross-linking, remove the culture medium and wash each dish 3 times with PBS, using 5 milliliters of PBS per wash. Remove the residual PBS as much as possible. For the experimental and no 4SU control samples, place the dishes on ice with the lid removed. Use a UV crosslinker to irradiate the cells.